Welcome to part 2 of chapter 8 on photosynthesis. So at this point, our photosynthetic organism, has harvested some light energy, and it's now going to build some sugars, carbohydrates, with that energy. So what products of those light reactions are used in this next step, this Calvin cycle? Right, so it's ATP and NADPH. So ATP for energy, NADPH for those high energy electrons. Some of the other choices you were given here either aren't products of the light reactions, or some are, but they're actually not used in this next step, the Calvin cycle. And so we can look at the whole process of photosynthesis here on this slide. Here we see the light reactions up here happening in thylakoid membranes, and then thylakoid space here. Producing some ATP from ATP synthase and passing those electrons to NADP plus to get NADPH. Those are what feed into this carbon fixation reaction. And this is just another way of looking at this process here as well. This is a figure from your textbook. All right, so carbon fixation, this occurs within the chloroplast, and again, we're using the ATP and NADPH, and if we wanted to build one glucose molecule, this is how much ATP would be needed and how many electron carriers would be needed to build that one glucose molecule. Now, glucose is not a direct product of the Calvin cycle. What you produce instead from the Calvin cycle is this 3-carbon compound, and then 2 of those can be used to make a glucose. So we can look at this in a couple of different ways. Here we see carbon coming in, carbon dioxide. So this is carbon fixation or carboxylation. Now we produce this 3-carbon compound here. We'll go into some more detail here shortly. And then reduction happens. What's reduction? Reduction is gain, right? Gaining electrons. Where are they gaining them from? NADPH. So these are passing the high energy electrons into these carbon-containing compounds in this reduction step. This also costs you some ATP, so some energy investment as well. This is going to produce one of those 3-carbon compounds. Well, you'd need 3 CO2 to get to one of these 3-carbon compounds, but it's there's a little more nuance to it than that. And then what's left in this is used to regenerate with some ATP. It's used to regenerate this starting molecule, RUBP. So let's look in a little more detail. We'll work our way up here. So here we're reminded that to get one of these G3P molecules, you need 3 CO2, carbon fixation, reduction, and one of these branches off here. And what's left, these other carbon compounds, as we'll see, are used to regenerate that CO2 acceptor, and around it goes. That's why it's a cycle. Here again, in a little more detail, inputting 3 CO2s. We're accounting for 3 carbons. Now we're keeping track a little more closely of the carbons. They're attached to this 5-carbon molecule, RUBP, one at a time. So here's that one that's attached. That's split, processed, energy's invested, high energy electrons are passed to it. So some rearrangements take place. And here one branches off. And again, to make glucose, you'd need 2 of these. So again, if you wanted to account for a whole glucose, you'd have to account for 6 carbon dioxides going in. What's left gets regenerated into this RUBP. It can be quite helpful to keep track of the carbons and where they go during the Calvin cycle. So we can look at this step by step. So we can start off with carbon fixation. And so that's where we're introducing our CO2. And let's imagine we're building a whole glucose molecule. So we're going to account for 6 CO2. Now they only go in one at a time, but we want to ultimately make one glucose, so we'll need 6 CO2. All right, so those are attached one at a time to that carrier, RUBP, ribulose bisphosphate. What catalyzes that, I'll get to in a bit. It's an enzyme called Rubisco. We'll look at that guy in more detail shortly. So let's keep track of our carbons. So here there's 6 carbons here. Each of these molecules, ribulose bisphosphate, they contain 5. So 6 times 5, there's 30 carbons accounted for there. Where does that get us? There's the carbon dioxide will be attached and it'll undergo some rearrangement here. And we'll ultimately end up in this first step with 12, what are called PGA. And that's phosphoglycerate. And each of those is a 3-carbon compound. So 3 times 12 is 36. So 6 carbons, 30 carbons equals 36 carbons, right? So, there's no carbons leaving this at this point. We haven't lost anything. Other ones haven't arrived from some random place, but we're just adding these and we can keep track of them one at a time. All right, so the next step is that reduction phase. So reduction, again, what's being reduced? These guys, this PGA, they're gonna get some more electrons. They'll be processed. That's boosting them up. Think of it like that, to a higher energy level. It's also gonna cost some ATP. And so we have our 12 PGA, phosphoglycerate, and I'll draw a little longer arrow here. We're going to produce from this our G3P. And so that's glyceraldehyde 3-phosphate. And I'll come back to that in a second. But I drew this arrow longer for a reason, because we're going to invest 12 ATP. So that's going to then, and we'll end up with 12 ADP as a consequence. We're also going to get that reducing power from NADPH. There's 12 of those too. Squeeze that in there. Let me extend that arrow down, and then we'll end up with 12 NADP plus as a consequence. So there's that ATP investment. Here's that reducing power. Now, 2 of these, 2 G3P, that's a 3. Imagine that's a 3. Those are going to be what's, say, invested to make our glucose. Glucose is a 6-carbon compound, and so you need to have 2 of these 3-carbon compounds to make it. And then the other 10 G3P, they're going to be used in the next step. So again, let's keep track of the carbons. There were 36 here. There are going to be produced, we still have 36. These are 3-carbon compounds. And then 30 of them get recycled. Meanwhile, 6 go off to make that glucose. That's what we're trying to make here. So our next step is regeneration. I'll just say regen. Regeneration of what, right? So it's of the RUBP. We're going to need that to go back to carbon fixation. And so where do we get it from? We're going to regenerate it using these G3Ps. So we got our 10 G3Ps. That's 30 carbons that we're accounting for. Draw a long arrow here. And that's going to produce RUBP. So let me write RUBP. This is a 5-carbon compound. We remember this from before. So how many of these can we get out of 10 G3P? We got 30 carbons to work with. We can get 6 of them. And look, back up at step 1, we needed 6 in this first step. So here's those 6 again. So we've regenerated that. This does take some energy investment. So here's the expenditure, if you will, of ATP. We need 6 of those. Remember, we needed 18 overall to make a glucose. So 12 up here, 6 here, and then that's going to, of course, give you 6 ADP. And we've regenerated that carrier that can go into carbon fixation. So that's the Calvin cycle. When we keep track of the carbons, it is probably also helpful to look at some of the videos of how this works, and you can see this in a little bit more dynamic way. So back to these steps now in a little more fixed fashion here. Again, you need to pay attention to what's going in. In this case, we're not building a whole glucose. There's 3 carbons feeding into this, so carbon fixation. Each one is attached to RUBP. This is catalyzed by Rubisco. Put a pin in that. We'll come back to that. Really important. And so this creates this very short-lived 6-carbon molecule, which is immediately split, and we produce that PGA. PGA, of course, then feeds into the next phase, which is reduction. So PGA will be, some phosphates will be transferred to it. Notice there's 2 phosphates on each one of these now. So this is phosphorylation that's occurring. Where do those phosphates come from? The ATP. And then this transfer of high energy electrons to this molecule here, bisphosphoglycerate. And now one phosphate off of each of those actually peels off now. So there's that inorganic phosphate that comes off. But now you have this high energy molecule. ATP energy has been invested in it. High energy electrons from NADPH have been invested in it. Now it can be used to make a glucose. Or again, it says other organic compounds, right? We're focused on glucose, but that's not the only possible outcome. Now, only one peels off at this point because we've only invested 3 carbon dioxide in this example. And so this would have to go around twice to get enough to make a glucose. But let's look at the last stage again here, regeneration of RUBP. So we've got, again, in this case, we've got 5 of these G3Ps, one of them peeled off down here. Some rearrangements take place. We need to invest some ATP, and then ultimately you build that RUBP again. All right, so that is the Calvin cycle and carbon fixation. Take some time with it, follow where things are going, and it really is fairly straightforward if you look at it step by step, very mechanistically. All right, but where is this happening? Now, we've already covered it. Do you remember? Where does the Calvin cycle take place? Right, so this takes place in the stroma of the chloroplast. So make sure you understand your chloroplast anatomy and where this is happening. Okay, so we've learned the basics of photosynthesis now. Let's start to bring this together with cell respiration again. And we can link this in what's called the energy cycle. And I've mentioned this before, this idea that photosynthesis is producing those things like sugars, and those are the starting points for cell respiration. So as photosynthesis is building these things, cell respiration is breaking them down. Or you could separate them between organisms. Photosynthesis in plants are producing these sugars, and then some other organism is eating the plants and then breaking those down by photosynthesis. But either way, the plants are doing both of these things, remember. In fact, approximately 50% of what photosynthesis is making just goes right into cell respiration in a plant. So, all right, we built these sugars, break them down, which you could feed in different places here. You see pyruvate actually being fed in here. Break them down and produce ATP. Again, so something else doesn't necessarily have to eat this. The plant is using it right away to make ATP. Other sugars may be used to make structural components of the cell, but most, or half at least, is fed into cell respiration. Okay, so with that in mind, where are the ATP synthase complexes in a plant cell? Okay, so if you took some time to think about it, you probably got this right away, or you pick something else at first and then maybe got there. Remember, plants have both chloroplasts and mitochondria, so they're going to have those ATP synthase complexes in both of those locations. Let's talk about a couple of things now that fit into this category of photosynthetic challenges. So one is too much light. Light seems good because you need that for these light reactions in photosynthesis, but too much, what can happen here is you run out of NADP+, because those electrons are being passed, and eventually you just run out. Now, we saw this before. If you get low NAD+, you can keep making ATP by cyclic electron transport. So these just go back here, pump protons, the electrons work their way around. This protein pumps protons, I should clarify. But some of them are passed to oxygen here. And so this is used to reduce O2, and you end up with this reactive oxygen species here. So some of these other pigments, so like xanthophylls, for example, help slow that rate of electron flow. And so that energy is dissipated as heat or released as heat. And so this can help slow that process in those cases. And so here's a question for you. Things like those xanthophylls and some other pigments can slow that electron flow. But how else does the plant protect itself from these reactive oxygen species when these electrons are passed to oxygen? All right, so plants produce antioxidants. And so these are things that you might think that are good to have in your diet that remove these reactive oxygen species. In us, it's the same in plants. Vitamin C, beta carotene are examples of this. And so we can look at these mechanisms that plants have to protect themselves, slowing down that electron flow. So like those xanthophylls here in the photosystem complex, that releases heat and light instead of passing the electrons along. So it slows things down. And then on the other end, when you do have these reactive oxygen species form, antioxidants can help take them away. Now, of course, you're losing some energy here, right? Losing some potential energy. That light energy is coming in and you're really trying to slow it down or almost lose it here by passing these high energy electrons off to something else. But it's better than the alternative, which is the damage caused by these reactive oxygen species. Here's something else that happens in plants. It's called photorespiration. So think of, we've been talking about cell respiration. What's happening there? We're breaking down organic compounds, producing CO2. So that's respiration. Now, when you think of photosynthesis, the opposite of that should be happening, right? We're taking CO2 from the atmosphere, the plant is, and it's building those carbon-containing compounds. So what's happening with this process of photorespiration is really the opposite of what you want to happen in photosynthesis. So in normal temperatures, for example, this enzyme, this is considered one of the most abundant enzymes on the planet. Think of, every photosynthetic organism has this enzyme. It's called rubisco. Here's the full name. And pay attention to this last part of the full name. So ribulose 1,5-bisphosphate carboxylase. This is what we've seen so far. This is carbon fixation, CO2 fixation, how we saw it, what was happening in the Calvin cycle. So that's that carboxylase enzyme activity. But it's also an oxygenase. So in different conditions, say like high temperatures, we'll see why that is, there'll be increased O2 concentrations in the plant cells or in the plant tissue. And what's going to happen is now this enzyme, instead of fixing carbon dioxide, it's going to incorporate O2 into RUBP, and it will produce carbon dioxide, which is the opposite of what's supposed to be happening. Now, again, normally the affinity for CO2 is still way more than oxygen, but if the concentrations of oxygen get high enough, it'll start using that over the CO2. And so in C3 plants, that's what we've been talking about so far. Why are they called C3? Because in that Calvin cycle, we were building that 3-carbon compound, right? Remember that? So that initial fixation of carbon, we build that 3-carbon compound, phosphoglycerate, PGA, in that first step. So in photorespiration, rubisco adds an O2 instead of CO2, and it produces these 2-carbon compounds we see down here, and that's phosphoglycolate. So 2 carbons there, and there you can actually see the oxygens that came from our oxygen here. So it's using oxygen, it's releasing CO2 instead of incorporating it into carbohydrates. So again, opposite of what should be happening. And we haven't produced any ATP or sugar as a consequence. So here's why this might be associated in climates where we have C3 plants that do typical what we've seen of the Calvin cycle so far. When it gets hot, say this is a cross-section of a leaf, they're trying to prevent water loss by closing these stomata. Remember these like sort of openings or mouths really in the leaf. So they close, that prevents water loss. That's good, I don't want to dry out. But what also happens is a byproduct of photosynthesis is oxygen. And because it cannot leave this tissue, it starts to build up as photosynthesis goes on. So more and more oxygen is present here. And then that will start being used by Rubisco. And here we see carbon dioxide not being able to get in. So, what does Rubisco have to work on? Eventually, even though it likes carbon dioxide better, if there's a lot of oxygen, it will start to use that. And so with C3 plants in hot climates, they can be basically wasting a lot of that carbon fixation that they've used to build sugars in the Calvin cycle as a loss to photorespiration. So this undoing of carbon fixation. So you can see here photosynthesis produces O2, incorporates CO2. What's happening in photorespiration? It's incorporating O2 and generating CO2. So really undoing this carbon compound building process. Here's just another way of looking at photorespiration. So again, oxygen being added to RUBP, that splits into, you do get this one PGA, but here's this other phosphoglycolate. It's a 2-carbon compound where the other carbon go, boom, broken off here as CO2. And look at this. We have to use ATP, and then of course we use ATP elsewhere in the Calvin cycle. And so this is, churning out carbon dioxide and using ATP. So you might wonder, well, why does this happen at all? This seems very counterintuitive in terms of being an adaptive benefit for a plant. So why hasn't natural selection weeded this out? Well, this could be what's considered an evolutionary relic. So photosynthesis and Rubisco evolved on a planet before there was very much oxygen in the atmosphere. So maybe there was no selection against incorporating oxygen because there just wasn't enough there. And so what we're left with is kind of an artifact as a consequence of this evolution that there's not strong enough selection against. So why might there not be strong selection against? Well, this might actually have an adaptive function. Maybe it's not just a relic, or it is partly, but it also has an adaptive function. So we know photorespiration limits the damaging products of the light reactions that build up when you don't have the Calvin cycle. So think about those, reactive oxygen species, for example. Some have proposed this enzymatic function might be important for incorporation of soil nitrates. So there may be a function of this, and maybe not just an evolutionary relic. Now, what we've seen so far is C3 plants that only have this one way to fix carbon. And so photorespiration is a problem because it can undo this process. And so when we're looking in tropical hot climates, this could be very severe. So what's happened? Plants have evolved different mechanisms to deal with this. And one kind of plant one type of plant are called the C4 plants. So think carbon 4. And what happens here is the plant will initially fix carbon dioxide, not right into the Calvin cycle, but in another cell, so spatially distributed here in a different place, fix carbon into this 4-carbon compound. So this is basically a temporary storage for CO2 in another part of the cell, or in another part of the plant rather, in these mesophyll cells. Those can be moved now into this, say, bundle sheath cell, and that carbon dioxide broken off. This will artificially raise the CO2 level in this particular cell. Now, what is this cell going to work on? Well, there's plenty of carbon dioxide because it keeps coming in this form. So even though the stomata on the plant are closed, that's okay. It doesn't need to worry about the gas exchange here because it's temporarily stored CO2 in this 4-carbon compound. Okay, so along those lines, why are C4 plants able to photosynthesize without photorespiration? Right, it's because they have that other step first. And we can look again a little more closely. Here's CO2 coming in, not directly into the Calvin cycle. That's down here in this other cell, but fixed into this 4-carbon compound. You can see a little more detail here, oxaloacetate, malate. That's moved into the bundle sheath cell. One of those carbons is chipped off, now that can be fed in here. And you can create a high carbon dioxide environment in this cell as these are moved in and these carbon dioxides chipped off, and off goes pyruvate, picks up another carbon dioxide, and round this goes. Now, that's not the only way that plants have evolved to get around this photorespiration. Some plants use this mechanism. They're called CAM plants, C-A-M plants, and that's crassulacean acid metabolism. And they use this to fix that carbon. That first step, okay, we're not going to plug it right into the Calvin cycle. We're going to fix carbon first in some other way. So think of, desert plants. So during the day, these plants, they close those stomata to retain water as much as possible. And so what's happening inside those plants is now carbon dioxide is being released. We'll see that in a second, broken off of that temporary storage molecule, and it creates that high CO2 environment within the plant. They don't need to rely on it from the atmosphere. Again, and this conserves water, right? Because they're not losing water with those stomata closed. You can see what that looks like in detail. So looking at CO2 absorption by this cactus plant, you look through the day, not really much CO2 absorption until you get to late at night. So it's cooler now, right? So water loss is less critical, and you start to see more and more CO2 absorption, which will, again, be fixed into that crassulacean acid, temporarily held there as basically a different compound. And then as the sun comes up and the day starts to warm up, CO2 absorption will stop. So here are those two ways that plants have around photorespiration. The C4 plants fix carbon in a different cell. So this is spatial separation. And then they'll move that organic compound into the bundle sheath cell and chip off the CO2. Here in these other tropical plants like the cacti that we saw, temporal separation. So this is not a different cell. We're just showing the shading here by different times. So CO2 is fixed at night when those stomata can open up and take it in from the atmosphere, and then fixed into an organic acid. And then during the day, stomata are closed to preserve water, and CO2 can be chipped off of these organic acids and now fed into the Calvin cycle at that time. Okay, so that's it for photosynthesis, and we'll see you next time.